Welcome back to the Deep Dive. Our mission today is to give you the ultimate shortcut into a really fascinating piece of ecological research. It's a paper titled Application of eDNA as a Tool for Assessing Fish Population Abundance. This work, led by Michael J. Spear and colleagues, came out in 2020, and it really tackles one of the biggest bottlenecks in modern conservation, I think. Okay, so let's unpack this a bit. The central challenge, and it sounds almost simple, is just counting things. If you're managing a big natural resource, you kind of need to know how many organisms are out there. But, how do you accurately count things like fish? They're mobile, they're hidden underwater, across maybe thousands of lakes, without, spending your entire budget.
It really is the foundational problem, isn't it, in aquatic resource management. We know that knowing the abundance, how many fish, what size they are, it's absolutely crucial. You need it for setting harvest limits, ensuring the stock stays healthy long term. But yeah, traditional methods are slow, they're known to be difficult, and honestly, staggeringly costly. And this is where environmental DNA, or eDNA, offers a potential breakthrough.
Right, for listeners maybe not familiar with it, eDNA is basically the DNA that every organism is constantly shedding into its environment. Think skin cells, waste, mucus, that sort of thing. The whole goal of this science is to take a water sample, analyze that shed DNA, and figure out not just if a species is present, but potentially how many of them are actually there, quantitatively.
Right. And what makes this particular deep dive so interesting is the species they focused on. The walleye, sander vitreous. This isn't just any fish, right? This species is the backbone of a massive, $2 billion plus annual fishing economy in Wisconsin. It's culturally huge across the upper Midwest. Exactly. And that economic importance kind of ironically makes the management challenge even bigger. Walleye aren't just in one big lake. In Wisconsin's ceded territory alone, you're talking about fisheries spread across more than 900 individual lakes and rivers. Each one really needs monitoring, but doing that traditionally. It takes a ton of time and people power for every single one.
Okay, so that leads us right to maybe the failure point of the current system. You mentioned the cost, the labor. Just how bad is this monitoring gap right now for walleye?
It's pretty bad, abysmal, really, because those traditional methods, we're talking mark recapture surveys, using nets, using electroshocking gear, they are so expensive and need so many trained people. Biologists right now can only monitor about 5% of those 900-plus walleye populations each year.
5%? Wow. Okay, that's kind of shocking, especially given the billions of dollars involved and the fact that we know some populations have been declining. Why is it so expensive? What does a traditional survey actually involve that makes it cost so much?
Well, you need a specialized crew, first off. Then expensive gear, like boats rigged with electroshockers or massive nets. You have to deploy all that stuff extensively across the lake. Then you handle the fish, you tag them, measure them, maybe take scales for aging, release them. Then you have to come back later for a second round of capture to see how many tagged fish you recapture versus new ones. It's weeks of really specialized work per lake.
Got it. Weeks per lake.
And that huge cost doesn't just mean few lakes get checked. It means even the lakes that are monitored might only be revisited every, say, 5 to 10 years. So you get this really infrequent data. It's poor quality in terms of timeliness. It just can't respond quickly if things are changing fast with the fish population. Management ends up being reactive, not proactive. And that's really the whole point of this study, trying to address that critical monitoring gap with something faster.
That sets the stage perfectly.
Yeah.
Okay, so Spear and his team set up this direct comparison. They looked at 24 lakes over 2 years. They contrasted those expensive traditional hands-on methods with this faster eDNA approach. Let's talk about the timing, though, because that seems like an important difference.
Absolutely. So the traditional measurements, kind of the gold standard for population assessment, those were done by the Wisconsin DNR and the Great Lakes Indian Fish and Wildlife Commission, GLAFWC. They did these intensive mark recapture surveys in early spring, usually May, right after the ice melted. And that timing specifically estimates the density of adult walleye, so individuals per hectare, and also the biomass, which is kilograms per hectare.
Okay, spring for the traditional survey,
right?
And then the eDNA sampling happened.
That was done later, in mid-July, months later. For that, they collected 1-liter water samples, pretty small volume, really, from 9 different spots within each lake to get a good average. Then back in the lab, they extracted all the DNA from the water and used a technique called real-time quantitative PCR, or qPCR. It targets a specific bit of mitochondrial DNA that's unique to walleye, and that tells them the concentration, basically, how many copies of walleye DNA were in each liter of lake water.
And I did notice reading the paper, they didn't actually use all 24 lakes in their main analysis. They took out 2, Bearskin Lake and Long Lake, saying they were extreme outliers in density or biomass. Doesn't that kind of risk biasing the results? I mean, you're essentially removing the toughest cases, which could make the eDNA look better than it is across the board.
That's a really fair question, and it's something you always have to watch for. But they were pretty upfront about why they did it. Bearskin Lake's density was almost 7 times the average for the region. Long Lake had biomass, mainly huge fish, that was also about 7 times the regional mean. Their argument was that including these extreme outliers would actually distort the relationship they were trying to find for the typical lakes managers deal with day to day. They weren't trying to build a model for like one bizarre reservoir. They needed something robust for the vast majority of those 900 typical fisheries. So yeah, the final R² applies to that typical scenario.
Okay, that makes sense if the goal is a practical tool for the common situation. And they didn't just compare eDNA to density, right? They looked at a bunch of other factors, covariates, things like water clarity, pH, lake size, depth, and crucially, the average size of the walleye themselves.
Exactly. They were really thorough. They tried to account for anything else that might affect how much DNA ends up in the water or how long it lasts before they could really be confident that the DNA concentration was reflecting the fish numbers.
That's great context. So now we get to the payoff, the big reveal. Given all the complexities, fish shedding DNA at different rates, DNA breaking down how strong was the connection they actually found between that liter of water and the results of those intensive netting surveys.
It was remarkably strong. They found a significant positive relationship, definitely. The initial simple model, just looking at eDNA concentration versus the walleye density from the surveys, that alone gave an R² of 0.62.
62%.
Okay, yeah, which means 62% of the variation in the eDNA signal was explained just by the number of fish per hectare. That's already pretty good for a messy natural system. But then their best model, the one that included not just density, but also the mean size of the walleye in kilograms, and used a statistical tweak called weighted least squares to account for variation within each lake. That model achieved a really powerful correlation, an R² of 0.81 across those 22 typical lakes.
Okay, 0.81. Now that number, R² equals 0.81, that's really the aha moment for you, the listener. Why? Because getting that kind of accuracy in the real world, in actual lakes, is huge. If you look at previous attempts to quantify animals with eDNA in natural systems, the average correlation, the average R², was maybe around 0.50. This result, 0.81, is getting close to the kind of accuracy people usually only see in super-controlled lab experiments, like in tanks, called mesocosms. Their average is about 0.82. So field results matching lab results, that's impressive.
It really is. They kind of cracked the code for walleye in these lakes, and the fact that the best models needed to include the average fish size, that gave a critical biological insight. It wasn't just about the number of fish or even the total weight. The analysis strongly suggested that for the same total biomass, say 100 kilograms of fish, if that 100 kilograms was made up of a few really big walleye, it actually produced less detectable eDNA than if that same 100 kilograms was made up of lots and lots of smaller walleye.
Huh. That's fascinating. Why would size change the eDNA output like that?
Well, it likely boils down to basic biology, physiology. Smaller, younger fish often have higher metabolic rates relative to their body size, and they also have a much higher surface area to volume ratio. So proportionally, they're shedding skin cells, producing waste, releasing mucus at a higher rate per unit of mass compared to a few big, older, maybe slower moving fish that add up to the same total weight.
Ah, okay.
Yeah.
More surface area, higher metabolism in the little guys.
Exactly, and that detail is absolutely critical if you want to build accurate eDNA models for other species or systems. You can't just rely on total weight. You probably need to factor in the size structure of the population.
So, okay, we've got this scientifically validated method, R² of 0.81. It accounts for fish size.
Right.
Great science. But what does it actually mean for the managers on the ground, the ones trying to balance fishing and conservation across those 900 lakes? Does this actually help them?
Yeah, that's the key question. Its primary use right now looks like being an early warning indicator, or maybe think of it as a triage tool. Managers often have to simplify things. They might classify lakes into really broad qualitative categories like, is the population fishable or is it non-fishable? Basically, is it healthy enough to support harvest or not? They need a quick way to figure out which category a lake likely falls into, especially for lakes they haven't surveyed in years. So the study tested exactly that. Can the eDNA assay classify lakes into just 2 bins, non-fishable, which they defined as 0 to 3.7 adult walleye per hectare, versus fishable, anything above 3.7 individuals per hectare. And when they did that comparison, the eDNA results achieved 82% agreement with where the traditional mark recapture surveys placed the lakes.
82% agreement sounds pretty good. But the paper also mentions a kappa coefficient of 0.4, describing it as fair to good. For the listeners maybe not familiar with kappa, what does 0.4 mean? Is fair to good really good enough to base management decisions on?
That's a fair question. Kappa is a statistic that measures agreement, but it specifically corrects for the amount of agreement you'd expect to happen just by random chance. So a kappa of 0 means your agreement is no better than flipping a coin. A kappa of 1 is perfect agreement. So 0.4, fair to good, means it's significantly better than random guessing. It shows a real, meaningful level of agreement for this kind of classification. Is it perfect? No. Is it useful for triage? Absolutely.
Okay, so better than guessing, useful for triage.
Right. That ability to distinguish 82% of the time between a potentially non-fishable lake and a fishable one, that makes eDNA valuable as a sort of firewall. Imagine a manager can screen, say, 50 lakes using eDNA for the cost and time it takes to do one full traditional survey. If that eDNA screen correctly flags the lakes likely in the non-fishable category most of the time, the manager can immediately say, okay, these lakes need our urgent attention. They can then prioritize those lakes for a full traditional survey to confirm, or maybe even more conservatively, just adjust harvest regulations immediately based on the eDNA warning signal. It provides crucial complementary information for that huge number of lakes, that 95% that currently get 0 monitoring in any given year.
But we definitely need to mention the limits here. Because while it worked well for 2 categories, the paper also found it didn't work so well when they tried to split lakes into 3 management bins, like low, medium, high density.
Correct. It performed poorly then, yes. The agreement dropped way down to 50%, and the kappa coefficient was only 0.2, which is considered poor. So this really highlights where the tool is strong and where it's not, at least yet. It seems quantitative eDNA at this stage is excellent for identifying those critical at-risk or low-density populations, the ones managers most need to worry about. But it doesn't seem to have the really fine-tuned resolution needed to, say, assign precise harvest quotas across 3 different tiers of abundance. It's more of a wide net surveillance tool than a surgical instrument for setting exact targets.
Okay, that makes sense. Now, shifting gears slightly to the caveats, when you compare a new method like eDNA to an old one like mark recapture, you always have to think about the uncertainty in both measurements, right?
Precisely. The traditional mark recapture estimates, even though we call them the gold standard, they aren't perfect. They have their own error bars. In this particular study, the traditional estimates had a mean coefficient of variation, or CV, of about 17%.
17% CV, so what does that mean in practical terms?
The CV is basically a measure of the relative variability, or the noise, around the estimate. So a 17% CV means there's a fair bit of inherent uncertainty just in that traditional population count itself. The true number could reasonably be 17% higher or lower than the estimate.
So the thing we're comparing eDNA against is itself maybe only, 83% precise. Why does acknowledging that 17% uncertainty matter so much?
Well, it makes the eDNA result, that R-squared of 0.81, even more impressive, actually. Think about it. They achieved a correlation that explains 81% of the variance, which is getting close to a perfect relationship, despite the fact that the benchmark they were comparing it to, the traditional estimate, has this built-in 17% wobble or uncertainty. It suggests the eDNA method is pulling a remarkably clear signal out of a naturally noisy system, even when the truth measure itself isn't perfectly sharp.
Interesting point. We also talked about the timing difference of spring surveys for adults, likely around spawning, versus the eDNA collection in mid-July, well after spawning. Did that seasonal difference, maybe different fish behavior or physiology, seem to affect the results much?
It seems like it didn't have a major impact, surprisingly, at least not on the models predicting adult density. They did look at whether including data on the young of the year fish, the age 0 fish that would be present in July but not so much in May, improved the models. But adding that age 0 data didn't significantly boost the predictive power for adult density. The strong relationship held up despite that 3-month time lag between the 2 sampling methods.
Okay, so we have this scientifically sound, rapid method. It correlates strongly with abundance, especially for flagging low-density populations. But what's the big piece it's still missing? Why isn't eDNA a complete replacement for putting nets in the water?
It comes down to the detailed demographic data. eDNA, as powerful as it is for abundance, gives you basically zero information on the fish themselves. It can't tell you the sex ratio of the population. It can't tell you the size distribution. Are they all small, all large, a healthy mix? It can't tell you their exact ages. And managers need that kind of demographic information to really understand population health, to model future trends, and to set truly sustainable harvest limits. So eDNA isn't a replacement. It's a fantastic, powerful, cost-effective complementary tool. It's designed to drastically expand the reach of monitoring across the landscape, filling in those huge gaps, not to completely take over the detailed work done by traditional surveys on key lakes.
That's a great summary of its role. This case study, than just one species, walleye, in this specific set of lakes, it really serves as a powerful proof of concept for the potential of quantitative eDNA, especially for monitoring these vast inland fisheries that are just fundamentally hard to keep track of. So wrapping up the main takeaway for you, the listener, eDNA looks like it provides a genuinely cost-effective, very rapid way to get a surprisingly accurate snapshot of fish abundance in the field. Mere lab-level accuracy, really. And that ability lets managers quickly assess the status of many, many more fisheries than before, acting almost like a firewall to catch problems early in those 95% of lakes that are currently data deficient.
Exactly. And thinking ahead, while this walleye study is a landmark, the next big step is generalizing these findings. To really turn this into a universal ecological toolkit, future research needs to dig deeper into the fundamental processes, the nitty-gritty of eDNA production, how much is shed, its persistence, how fast does it break down under different conditions, and its capture, how do we best sample it. We also need more work on standardizing the protocols so results are comparable across studies and locations. That underlying scientific rigor is what will unlock its true potential for broader conservation.
Which brings us to a final thought. Traditional methods we rely on now, like electroshocking, they were actually met with a lot of skepticism when they were first introduced maybe a century ago. People thought they were strange, maybe harmful, maybe inaccurate. So considering that history, it makes you wonder, how quickly will the perception of eDNA shift among fisheries managers? How long until it goes from being seen as this novel, maybe slightly experimental secondary tool, to being viewed as an absolute necessity for managing these complex, vital ecosystems? Something for you to think about until our next deep dive.